Hello everyone, welcome to the hands-on tutorial on the protein ligand molecular docking interaction analysis. And in this tutorial, we are not going to use any kind of software or any kind of tools or any kind of other third-party websites. We are only going to use Google Colab, and the entire pipeline is designed to do the molecular docking analysis of protein and a ligand. The materials of this project and the scripts and the entire pipeline will be available on the course page of the dlxbio.com, and you can simply run the entire pipeline. Just you need to change the protein and the ligand of your interest, and you need to run the entire pipeline, and in a few minutes or maybe less than an hour you will get the results of the protein docking interaction. So to continue with this, we'll like to see what this pipeline basically covers. So basically this is the molecular docking workflow in which a protein and a ligand will be downloaded, and the interaction will be done in the Google Colab, and the results will be generated. So this process has 19 different steps which we will be checking one by one. In the first step, the setup will be arranged, and the system information will be checked, like what is the disk space, RAM and the CPU and the version of the Python that we are using. Then we'll be connecting our Google Colab to our Google Drive, and where we'll be creating the multiple projects and the folders, and the structures will be sorted, and it will install the required softwares, and it will verify the installations, and later we'll be coming to the part 2, where we'll be downloading the protein of the interest and the ligand molecules. Further, part 3 will download the protein, and sorry, in part 2 we'll be selecting the protein molecules, we'll be assigning them to the system, and then in part 3 the exact molecular structures will be downloaded from pdb, and also they will explore the protein and the different chains, residues and the characteristics of the protein. Then we'll be do the pre-processing, where we'll be removing the water and other ions or other things, and further we'll be doing the validation of the protein using the different plots and the diagrams, and the protein will be prepared for the docking analysis. uh, on the other hand, we'll be downloading the ligand from database like PubChem, and it will be converted to SDF to PDB, and then we'll be doing the ligand verification, ligand validation, like molecular weight, HBD, HBA, and rotatable bonds, and TPSA, log P, smiles, and others. This will be followed by pocket detection and the grid box selection, and further the docking interaction will be performed, where the uh, prepared protein and the prepared ligand will be analyzed for the docking analysis, and then the output will be given. This will further follows the visualization of the docking for identifying the best poses, and identifying the interaction analysis of residue residues level, and then there will be the validation of the docking analysis, and the results will be exported in the form of the images, pdb, pdbqt, and csv, and summary tables, and we'll also generate a pdf which can be served as a publication ready format for our analysis. So to move on with the first part of the project, where we'll be doing the Google Colab setup, and then we'll be checking the system configuration and all of the things. So basically this output will be something like this, where like it will tell us what is the approximate RAM, and what is the ligand preparation size, what is required, and what are the configuration of the system. So let's start with this. So we'll go to our Google Chrome, and this is the Colab notebook, this IPNYB file. So this is basically available on dehelixbio.com. Here in the course page, you will go down and you will get the protein ligand using Colab notebook. This is the course, and you can open the drive. So basically here you have the course materials for all the courses, and you can find out which is the course you are currently checking out. And I'll also share the link in the drive sorry YouTube comment section, sorry, the description below. And so to start with our docking, so we'll open the Google Colab notebook, and here you will see this is the number of the codes which I've already designed here. And we'll start with each code one by one. So you can either do run all, and otherwise you can do one by one. So basically this is a very entire long process, is very long, and there is more than hundreds of this panels. So make sure you first connect this to your operating system, and it will assign you a space of RAM of around 12.7 GB, and 107 GB approximately disk space. So to start with, we can either enter run all. So right now we'll select run all, and the first process has been initiated, and it has been subsequently running all the processes. So it is asking you to connect with the Google Drive. So you should connect with Google Drive so that you can. So basically, you should log in with your Gmail ID, and you should have some sufficient amount of space, at least 20-50 GBs, so that you can save your final output files. So you should connect to this Google Drive, and after a couple of processes it will automatically get connected to your Google Drive, and it will save all your outputs there. So make sure you select here, select all of them, such that this Colab can basically create a folder in your Google Drive and access the files in that folder, and also it can edit or modify the files. So basically, when you are converting PDB to SDF or SDF to PDB, any file format conversion, docking interaction, or you are writing, you are saving a new file like CSV file or any PDF file, so it should have the access to basically edit and uh, modify all your files. So once the process has been done, we can see that the first is the Google Colab information, and it's showing the operating system is the Linux, and machine processor, all these things, cores, and RAM is assigned, and then the disk space, free disk, and used amount. So all these things are being shown. And the GPU, there is no GPU. So basically, when you are doing large scale molecular docking studies or maybe high configuration things, so might need GPU, so which we will be covering in subsequent tutorials. So right now we have imported the Python packages, so NumPy, Pandas, and Matplotlib and all this. So these are the codes. So basically we need to understand what we are doing exactly. So otherwise you will simply run the codes and you will generate it, but you can't really write in your project or in your research what is basically what you have done basically in your uh, entire project. So this is the entire workflow, and uh, so let's get back to our presentation, where we have already done all these things. In the subsequent step, we'll be mounting our environment to the Google Colab, which we have already done. Then we'll be installing the docking softwares, and as I have already uh, mentioned, and test the 3D viewer with a dummy molecule. Uh, After that, will be for part two, certain molecules will be downloaded. So let's see what is happening in our Colab. So uh, step three has been done, and then verify installation, test the 3D viewer. So you can see the molecule here. You can expand or you can rotate it. So this is the dummy molecule, and you are basically validating this. So step 5 is download the protein, and here will be downloading the protein. So the protein structure has been downloaded successfully, and then we'll be checking what is the basically this protein has the information. So these are the display first 30 lines of the PDB files. So it will show all the different information about the protein, like the organisms, and then Homo sapiens, and the like name of the protein, and expression system, all these things. So you can basically write this in your description section of the protein. And the statistics, like residues, the 404 atoms are 2506 residues. And this is the overall structure, like our first real protein visualization. The protein should look like this. So you can rotate it, you can check this. So this almost works like your this docking tools, where you basically visualize your protein and perform the docking. So only difference is you will be doing multiple tools. Here entire process will be done in a single pipeline. So if it is very convenient for you, you can use it, because this is a very simple project. In the upcoming tutorials, we'll be dealing with multiple projects, and I'll try to simplify the pipeline as easiest as possible, so that you do it in a minimum number of steps by saving the time and your efforts. So now count the standard amino acids and the water and the heteromolecules. This is the step where you basically do this. So I think all these steps are not required for everyone, but maybe some of the informations will be useful for someone, like element composition and all. So this is the entire pipeline. And again here, this is the protein in multiple styles you can see. So you can see here there is another molecule inside this protein, and we can rotate this protein, and it will show the information related to this. So ATP can be visualized separately. So this is the ATP molecule. Now we'll be showing the protein surface. This is the surface of the protein, and then show only ATP. Here this ATP molecule is only shown. Then finding the ATP coordinates, so it will locate your ATP coordinates. So maybe this is not useful for everyone, but someone will maybe finding this useful. So I have made this entire tutorial for a large population, so maybe you can check out what is really useful for you and what is not. So some of the ATPs which are the ATP is bound to some of the protein residues, amino acid residues, and you can basically find these here, and it will highlight the binding sites, and it will show the output like this. You can zoom in and zoom out, and you can clean the protein, where you will remove all the water molecules and all the other atoms which are not required. So let's see what is there in the uh, this thing. So part 2 is done. Then part 4, where we will be exploring the protein and doing the cleaning of the protein, like water, ligands, ions, and alternate locations. So we'll be basically cleaning the protein, and it will generate output like this, where they will give the protein chain a and the number of amino acids and the ATP, and whether any other ions are present on it, and it will clean it. And so here we are in the step 4, where we'll be visualizing the ATP and all. Then in part 5, the cleaning process will be conducted, where it will this is the entire workflow for the part 5, where we'll basically clean the protein, like it will retrieve the original structure and identify whether everything is present, and it will show the water molecules, and it will visualize before and after of the cleaning step, and remove the ATP, validate ATP removal, and magnesium ion, check magnesium ion, compare all the structures, and all this you can check it out. So then we'll be going with the ligand downloading, and it will give the informations regarding the ligands. So basically it will be downloaded from PubChem in the form of SDF. It will try to validate the molecule, it will read it, generate the 2D structure, and it will convert it into PDB format, so that it's ready for docking interaction, and it will generate the output in this manner. So let's go back to our uh, Colab, where so basically in this step we are visualizing the water molecules of the protein, and then we'll be removing these water molecules. Okay, visualizing ATP and the water molecules together. Further, we will be removing the waters, and remaining water it will be showing 0. Then visualize after the removal of the water molecule. Further, we will be removing the ATP. ATP has been removed successfully, and it will be validating then. Then there will be magnesium ions, and then after that it will validate after ATP removal. So you can see there is that tiny molecule of ATP is no longer present in this protein structure. Now it will compare all the protein structures. So basically you can see what is basically the previous one and the final structures. Original has one ATP and one magnesium, and there is number of water is 294 in the structure, and the final one has no water, no ATP is 294, means it has cleaned the protein. Then it will check the protein quality, protein loaded successfully into the PDV fixer, and it will detect the missing residues, and after that there will be this like heavy atoms and terminals, all these things will be generated. So it will add the hydrogen at a level of the pH 7.4, so which is basically the optimum pH of our body, yeah, and not the digestive system but the entire body. So it will save the prepared uh, receptor, and after that it will compare these proteins. So you can see the summary of the protein preparation, and it will visualize the receptor. So basically this is the protein is the only receptor, and then we'll be working on the ligand. So you can see this is the cleaned protein. Further, it will download the ligand. This is the next step where we'll be downloading the ligand. It will generate the ligand information, and ligand downloaded successfully will be the output. And there will be ligand name, like the name of the ligand is staurosporine, which has the CID of 44259. So basically this is the dummy tutorial, so this is why we are using this. So otherwise you can change the name of the ligand, and otherwise you can simply give the URL of the ligand, which you can do from literature review. So it will download this, and then it will validate this. So it will read the ligand, generate the 2D and the 3D structures, and then convert to PDB. So it has done all the work, and it has saved this ligand into the 2D format, and then it will give the 3D format of this ligand, which you can visualize. Further, it will validate the protein. So here it will see that you can see that this protein name has been validated. So once this is done, there will be some structural statistics. So basically the chains, number of residues, atoms, and B factors, and which are the most common amino acids. So it will also generate you the validation summary, and you can save this in your like it will automatically save it. You can use this in your research proposal, research paper or article, or even in your presentation. So after this it will show you the structure, and then further you can visualize the flexibility using the p factor. Maybe this is not much useful, but if someone is looking for this, this is available in this pipeline as well. Then it will validate the active sites, and you can see it has generated the 3D image, and you can use this for your presentation purpose. So wherever this molecule will bind, it will show this kind of configuration. Then it will do the ligand validation, so it will show the properties and the values, where molecular formula is given, and the molecular weight, mass, and other factors has been given, number of hydrogen donors, hydrogen bond acceptors, uh, rotatable bonds, ring count, and all these things are there. Then it will follow the Lipinski rule evaluation, so that it is basically so violation is 0, so it is an excellent drug like ligand. So there are 5 rules of Lipinski which should be uh, followed by any drug molecule to be called as a drug. What we'll be doing further is the ligand visualization. So it will show the structure of the ligand both in the 2D format and the 3D format, and you can see this here. And then it will check the pocket detection. So let's get back to our PPT. We'll see what is basically happening. Protein validation is done. So basically this workflow is like it will read the protein information, the chains and the B factors, and then it will visualize the protein quality flexibility using the B factor and the active side of the visualization. The ligand validation, we have, as we have seen, it has generated the 2D and the 3D structures after this. And then you can see the protein detection. There is the interactive pocket visualization, and then the pocket surface visualization, and it will also show the residues to which the binding will be taking place. So this is the part 9 which we'll be seeing again, and then in part 10 there will be grid box selection. And let's go back to our Colab. So in this part 9, this is the pocket detection, so it will give you the residues in this format. And there will be interactive visualization where you can see the where the protein and the docking and the drug docking will be occurring. And further you can see the pocket surface visualization in the 3D format, and this you can also add in your presentation. It will also define the grid parameters, like this will be in the form of this table. Further it will show the grid center. So here you can generate the grid centers and visualize the docking box. Here you can see the center is there, so x-axis, y-axis and z-axis, and the surface of size of this uh, interaction will be docking space will be given. The next step is the protein preparation for AutoDock Vina, and this final receptor validation will be occurred, where the output will be generated like final receptor validation. It will again validate the protein so that you can understand whether it is ready for this interaction analysis or not. And then it will generate the receptor preparation report, again the same things, and it will understand the PDBQT in the next step, where it will basically see whether the uh, conversion or the preparation of the protein is successful or not. Then in the part 12, the ligand preparation for docking. So here again it will prepare the ligand, and it will give a visual representation whether it is ready for the docking analysis or not, and it will prepare a report like what are the properties of the final structure. Then the part 13, the complete docking environment will be there, and it will basically bring both the files together. So in a single environment, it will bring the uh, protein and the uh, ligand together. And so here you can see that this is the finalized receptor, this is the interaction, and the features of this interaction can be generated with the output. In the part 13, the complete docking environment setup uh, will be there, like the folders and the paths will be on there. So you can see that the receptor PDBQT, ligand PDBQT are successfully added, and then all the other uh, required setup are done. So basically in this process there will be multiple these things, libraries will be called, like Vina configuration will be done, ligand torsions and pocket centers, all these things will be done by the system. And so once the all the files are uh, put in a single place, it will perform the uh, preparing of the receptors and the ligand, and further it will validate whether they are in the ready configuration, and it will provide the results in somewhat like this format, where it will show whether they are existing in the same place, they have the size, their atomic configuration, the root, branch, and all other factors. These are the some of the informations that you can generate with this part. And then it will give a visual check of the input structures, and it will generate the structural configuration of this protein and the ligand. Then it will install the AutoDock Vina, and once the installation is finished, it will take a few seconds to do the installation, and it will do the verification of the installation whether it has successfully installed. And then we'll come to the part 14, where basically we'll exactly run the Autodoc Vina in this model. And it will take some time. In this configuration will take a lot of time because it is actually performing the docking analysis, and it will basically generate the output in this form, and it will show that the docking has been successfully completed. Then it will validate the docking files, and the these files like this docked pdbqt file exists, and its size and the output of the terminal. Then it will read the logs of the output, and it will show the different properties that has been done with the on docking. There will be certain docking scores like this binding affinity in the kilocalorie per mole, then RMSD bound, and RMSD lower bound, and RMSD upper bound. So this will be generated, and the best docking pose results and the scores will be reflected here. Then it will generate the summary report and the docking summary like protein, ligand, protein name, ligand name, and the docking grid, grid space, grid size, and the binding affinity like negative 14.9 kilocalorie per mole, number of poses, and the status like whether docking has been done or not. Then you can visualize the docked complex. So here you can see number of the parameters here. 18 molecules have been converted, and you can visualize in this panel so where exactly the docking is taking place. So once it is done, uh, okay, you can also visualize in this way in different kind of display mode. There is the line display and the ball and stick display. You can basically customize this, and then you will compare the original versus the ATP binding sites here in this step. So where basically this ATP is binding and the docking part is binding is happening, and then you will see the visualization summary. What are the things that are conducted here? Then we'll move to the interact. Uh, Further moving to the part 16, uh, what we have in part 15 is we have visualized the docking surfaces, docking grids, and in part 16 it will give a report of the interaction, and it will basically classify the interaction and the report of the molecular interaction with the residues, residue number, and the minimum distance. And as we can see here, this is the interaction of the part 16, and the residue number, and the minimum distance. It has generated the summary, and then it will also generate the image like this, the 3D image. The interaction summary will be displayed, and further it will classify the contents like these are the residues like aspartate and lysine, all these residues with the residue numbers, and the minimum distance and the type of the interactions. Further, it will also give a visual representation of the interaction that has been conducted here, and it will give the summary of the interaction, which you can use in your result section. So basically the next part is the validation of the docking. So in the docking validation, it will first generate this kind of visual infographics, and it will also give a another visual representation of the pocket occupancy, and it will validate the pocket occupancy, and then it will give the matrices of the validation, like the best binding affinity, which is basically here negative 14.19 kilocalorie per mole, number of poses, and then the whether the protein is prepared, ligand is prepared, grid is validation is done, and whether interactions are detected. Then it will give the docking validation report and the overall validation score. And in the final part, in the part 18, we will see how it has exported the result. It will export the result in the form of images, PDB files, PDBQT, and the CSV, and also the summary tables. So you can see these are the docking table. It will be exported in this way. The docking summary will be given, and the conclusion of this study will be displayed, and it will also basically give suggest us what is the pipeline stage, and what is the status of that pipeline. The final your uh, interaction analysis will be saved in the different folders, and you can see this is how the folders are designed. In the report section, you can find the final docking results and reports in the form of the PDF. uh, let's see what is going on here. The results will be exported and the files will be exported. So let's see where the files exactly are. So here you can select the file section, and select the, I think, sample data, and we'll see whether, so no so once you are connected to the Google Drive, it will show where exactly your things are there. So here you can see a file named protein ligand docking tutorial, where all other files are saved. And if you go to the reports, you can see what are the different reports, like docking summary.txt, ligand preparation report.txt, and receptor ligand report. And it will also show the results. uh, Here there the visualization summary, and also final results. You can see this docking and then images. In the docking section, you can see this the PDBQT file. In the images, you can see the images that are generated here, and then further on the reports, you can see the PDF file. So if you double click on that, it will I think download it, and you can see the results here. So this is the protein ligand docking report, which you can download from this, and it will show all the parameters and the information that has been used in this study. And you can see the docking score of negative 14.2 kilocalorie per mole. It indicates that it is a highly favorable interaction. The ligand remained inside the predefined binding pocket and established close contact with this few amino acids, aspartate 141 to lysine 33. So overall conclusion, it will generate the docking experiment demonstrates a stable protein ligand complex suitable for further molecular dynamic simulations and other experiments. So it will generate a PDF report like this, and also there will be these images related to this study, which you can also access from your Google Drive. And what else we can see is that final docking report from here. This is the final docking report, which is basically similar to the PDF what we have seen, and it's also in the form of the text format. uh, there is this docking summary.txt file which basically shows what is the status of the docking, and these are the basically the uh, associated files with your study which you can use for your research article or your publication material. So once the entire pipeline is completed, it will give all the things as successful without any errors. And if you are kind of getting any kind of error, you can just comment below, down the video. We'll try to fix that errors in that file. And also you can use any AI tools or ChatGPT for fixing the errors yourself, and you can also keep this pipeline in your GitHub repository. So our GitHub repository will be active in after the video is done, and you can access the files from there as well, and you can run in your own system. If you don't have high-end system, you can use this Google Colab. Otherwise we can use the Jupyter notebook for running some more complex studies. We'll look into other studies in the upcoming sessions. And as of now, I think we have completed all the uh, steps of this pipeline without uh, getting any errors. So to summarize this, we have gone through 19 different parts, out of which we have basically set up the Google Colab environment. We have installed multiple libraries and required softwares. We have created different folders and imported different files, particularly the protein and the ligand files. We have validated the information, whether all of them are true, and whether they are suitable for docking and interaction analysis. Further, we have uh, prepared the protein and the ligand for our docking analysis. Then we have run the docking output, then we have done the docking analysis of the protein and the ligand, which basically is the most time consuming part of this process. Otherwise the entire pipeline basically runs within less than 10-15 minutes. And since the entire pipeline is completely designed, so you don't need much time until or unless you get any errors to fix this. So we have also seen how we have summarized the interaction analysis, and we have validated the docking. Finally, you have exported the results, and in the form of the PDF, PDBQT, CSV, different images, and also summary tables. So you can simply try out with different proteins and other small molecules, natural compounds, or which basically act as the ligands. And you can basically do multiple different kinds of projects, which is basically a small short project for school level, I mean college level students, and you can use this for your publication as well. So thank you for visiting this video, and once again I'd like to show you how you can access uh, these files. So you can simply go to dehelixbio.com, and there you will get this course page, explore courses, where we have already given all our previously free courses. Below your in your previous course materials, you will get this part, protein ligand docking, using the Google Colab notebook. And once you open the drive, you can simply access this uh, file, and you can simply open this and run this interaction by connecting it to the suitable servers. uh, so I think that's all about this video. Try it out yourself, and let me know what other projects you are looking for, what other analytical uh, guides or tutorials you are looking for, and maybe we'll try to bring some of those tutorials in the upcoming videos. So thanks for getting in touch with us. Stay connected. Okay, you can subscribe our channel if you find this video useful, and you can refer this to others, whether they can use this pipeline, and also let us know your feedback on this video. So thank you so much for staying with us. uh, See you in the upcoming videos.